problems by using chemical techniques. And it's that spirit that I worked in all my career, as you will see some of the steps in my career. But it was actually Tom who kept me fairly sane in the lab by talking to me and keeping me calm at times of stress. And because a young postdoc gets quite stressed things. But so I regarded Tom as actually an important mentor in my life as well. So thank you, Tom. Now I spent most of my career at 1975 to 2012 in this building at the MRC, the old building, which was actually opened in the 1960s by the Queen. And my lab was up there, the chemistry lab, which was right next to the fire escape, just in case there were any problems. We never had any problems, but we didn't have to go down the fire escape, but it was right there. But in 2013, we moved into a new lab, actually, on the other side of the campus. It's a wonderful new lab, and that we've been there ever since. And anybody who's coming to England, please come and visit this lab, because it really is an amazing place. I'm now just about to retire, although, as you will see, I'm still going to be very busy, and I shall still maintain an office here, but we're also starting a company, as you'll see. Now, actually, I was, as Marcia says, one of the chain gang making oligonucleotides. This is a picture from 1976, and you will see, where the Gobind, you'll see Rama at the back there. Rama, who's actually here. Now, it was actually Rama who taught me my oligonucleotide synthesis at the time. He was there at the bench right next to me, and I thank Rama. It's wonderful you're here, because you taught me all I needed to know about how to make an oligonucleotide by the phosphodiester chemistry method. And I actually made a sequence in the promoter region here, GGAAGC, GGGGC, actually ingrained in my heart. It took a whole year to make that first oligonucleotide, and then I stayed for a second year to make a second oligonucleotide. So in those days, it took a whole year to make an oligonucleotide. That's why there were so many of us on the chain gang, because it took a long time to make this tyrosine suppressor tRNA gene, but it was functional, as Tom said, and that was really very important. In a series of papers, this is the second gene, the alanine gene, tRNA gene was the first one, but this was the first functional gene in 1979 in the series of J. Bowell Chem papers. Very formative period. I made great number of friends and colleagues who have now remained, and Parul is here as well, who was in the lab at the same time, it was a great time. But I obviously moved on, but to Cambridge, and over the period, as Tom said, we had many reunion meetings, and this was the first one that I was one of the co-organizers, along with David Jones, who's actually there. It was a shame that he couldn't come here. He was supposed to be here, and Dick Walker was on the back here. Gobind and Esther in the middle here. And there were lots of people you might, Gordon Tanner was here, I think, and a lot of people you might recognize. Rama was here somewhere here, and Hans Gross, and a lot of Bob Wells, a lot of people at this first meeting here. It was quite an exciting meeting. Gobind also was able to have a reunion with Lord Todd. This is Lord Alexander Todd here. This is the last time that they met, as far as I'm aware. He was just closed his lab now, and he was now a emeritus scientist. But it was a chance for him to meet up, having worked with him as a postdoc some years ago. And also at this meeting was Kell and Ruth Kleppe. Now, Kell Kleppe actually should have got a Nobel Prize with Gobind, and Gobind a second Nobel Prize, because actually the principles of PCR, of polymerase chain amplification of DNA using regular polymerases, was actually Kell and Gobind's idea at the time. Kary Mullis developed the thermonuclear stable polymerases, but actually the original concept was Kell. But that's another story that I won't go into. But it was we had lots of very important people at this conference in 1985, and of course we've gone on ever since to other ones. Now, when I started in Cambridge in 1975, I obviously, I decided to stay in oligonucleotides, and I was hired to actually use a new solid phase method. My mentor here was Bob Shepard, a peptide chemist, and we developed methods of solid phase synthesis of DNA using his new polyamide support, which he thought would be freely permeated by polar organic solvents like pyridine and DMF. And so it proved to be. And I used Gobin's phosphodiester chemistry on a solid support and developed the technique of solid phase synthesis. And this was fairly manual, although we used actually a modified Beckman peptide synthesizer to deliver the solvents. It was just purely delivery agent. And the cycle time now was about 24 hours to add one nucleotide. It was quite a lot of work actually in those days, but nevertheless, and there's a series of papers here I published with Bob Shepard here, but nevertheless it was a significant improvement in time. Now it's fairly manual, and Arama recognized the dry box actually that we used to do phosphodiester chemistry, where I still use the dry box in that initial method, and I actually activated the nucleotides with triisopropyl benzene sulfonyl chloride, just as we used to do, and blew them into the reaction vessel. The reaction vessel was held in a little glass, sintered glass funnel, a little glass reaction chamber. And later I used a switching device to switch manually. I modified it from the Beckman synthesizer so I could deliver it by gas pressure. And it had a timer and what have you. So then, after purification, both at the deprotected and protected and deprotected stage, also quite a lot of work. But it actually now took an assembly of about 1 to 2 weeks to make an oligonucleotide by 1979, rather than the 1 year to make an oligonucleotide. So this was already a significant advance, including 1 week for purification. So that was an improvement. This is my colleague Bob Shepard, whose lab it was, and there was his peptide group. But I was afforded little space here, and actually my first assistant was Mohinder Singh, who was actually from Chandigarh. He's actually retired to Chandigarh, and I stayed the last few days with him. He was my assistant for many years, from 1979 to 2003. And there was a couple of other people. There's me at the back there, and Richard Titmus and Hans Mattes were my first postdocs. But these are the other peptide group. It was in the days when we were allowed alcohol in the lab. Nowadays, health and safety don't allow us. This was a party there. But we switched to the triester chemistry method at that point. There was a new method of oximate, a cleavage of the chlorophenyl protecting group at that time that Colin Rees introduced. And this inspired me to change to the triester method of chemistry, using first of all a polyamide support and again delivery by the Beckman peptide synthesizer, later a more rigid Kieselguhr polyamide support. And now we started to pressurize the bottles with nitrogen pressure and pack the resin into a small column. And the small column actually is shown here. These are little columns here, and the resin was packed into. These are 4 columns in parallel. Had a switching device and bottles. And the idea of using pressurized bottles came from Marv Carruthers, who visited my lab in 1981. And suggested, why don't I switch to gas? And in fact, we never looked back. We used apparatus from a small company called Omnifit, which provided the bottles and all the plastic tubing and things like that. And now developed a cycle time of only 1 hour rather than 1 day, and using a long chain alkylamine controlled pore glass support. We were able to synthesize quite rapidly. So the cycle of deprotection afterwards, followed by purification. So now we could assemble very quickly. In fact, my last postdoc at that time, 1984, 1982-84, Brian Sproat, assembled a whole gene by himself on this apparatus, this manual apparatus, using 4 column, 23 oligonucleotides up to 22 nucleotides long, synthesized 4 at a time. And the whole synthesis was done, including the cloning and ligation into a vector, M13 vector, and I did that with Mahinder Singh. And the whole thing was done in 3 months. So that's compared to a whole army of people just 8 years previously in Gobin's lab to make all the oligonucleotides and the cloning. So already now things are rapidly improved in time. Actually, we trained quite a lot of molecular biologists came down to my lab from the other parts of the building and actually assembled their own oligonucleotides using this little manual synthesizer. And in fact, the company did rather well. We didn't make any money, but the company sold hundreds of these all around the world, including in India here at the time. So it was actually quite an exciting time seeing our triester chemistry going quite nicely around the world. However, things were changing. Marv Carruthers had developed a wonderful new phosphoramidite chemistry in Boulder, Colorado, and also using a glass support. And he founded the company Applied Biosystems, and that chemistry was just slightly better than the phosphotriester chemistry. It gave a higher cycle yield, and therefore you get a longer oligonucleotides quite quickly. And because of the starting of the company Applied Biosystems, very, very good engineering, it was clear that the race to make oligonucleotides and automate it was actually run by Marv and his company. We switched to the phosphoramidite method in 1985 and never looked back. This is me with some hair at that time, posing in front of the machine. I didn't do it. It was a whole series of people. We formed an oligonucleotide synthesis service in 1985, and that lasted to 2010 with a whole of these sorts of people. And we could make whole genes, and this was actually a collaboration with Pushpa Bhargava in Hyderabad, making a seminal plasmin gene in 1987, which we made and expressed the protein and did some protein engineering with. And this was, of course, done quite quickly. The oligonucleotides we made now in just a day with no problem at all. So having the oligonucleotide synthesis was now essentially solved, and the machines now could churn out these oligo-DNA, these DNA fragments. So I turned my attention to RNA synthesis at that point, solid phase RNA, and also some other end chemistry. So with Chris Christodoulou, we did some oligo-ribonucleotide synthesis using an FMOC method at the time. This was quite popular for a very short time in the late 1980s, and people were starting to look at this quite clearly. But then the Ogilvy-Salal chemistry came on board, and we started to be able to buy monomers. So we dropped that in sort of Ogilvy-Salal chemistry. But we also we made non-radioactive labels to the end of synthetic oligonucleotides, and did some in situ hybridization with Piers Empson at that time. And we used an FMOC linker. In fact, we believe we were the first linker to be made at that time and published in 1986. And Sudhir Agrawal from Allahabad, he was just a postdoc at the time, did this work, and now Sudhir went on to become a CEO of Hybridon and then Idera, and became a really high-flying executive and wonderful chemist in this company, and he's just retiring at the moment now. And Sudhir tells me that if we had patented at this time, we'd have made a lot of money, because this was the basis of linkers onto solid support of our diagnostics for many companies used. But at that time, we were not doing patenting at all, which is a great shame. But never mind, we provided it for the world, which was fine. So we did a lot of labeling at that point. But we also started to use RNA in various other methods, and particularly looking at the hairpin and the hammerhead ribozymes. It was actually Aaron Klug who asked me to start thinking about working on the hammerhead ribozyme. He wanted to actually crystallize it, and so we started to look at that. And I decided to do some chemistry on this. And we first of all worked on the hammerhead ribozyme, and we were the first to show using configurationally defined phosphorothioates. Phosphorothioates came from Fritz Eckstein's lab, to study the mechanism of cleavage of the hammerhead ribozymes, and showed it actually was, as predicted, an inline cleavage of the hammerhead ribozyme, and then went on to the hairpin ribozyme and did some work on the structure and putting in some base analogues, looking at the purine functional groups, and then later doing some cross-linking here, looking at the structure here. We worked with Frank Zahler. Eric Westhoff did some modeling for us, and Fritz Eckstein provided some materials for us. And we actually, the predicted structure of the hairpin ribozyme turned out to be very close to that which when the crystal structure came, so we were quite kind of happy about that. And we also looked at aminoglycoside antibiotics and polyamine spermine, showing we could reduce the metal ion requirement for the hairpin ribozyme cleavage. So that was quite an informative time and quite exciting. But a change of pace occurred in 1988 when I was looking for a more a mammalian system to work on. I thought it was time to go from, I'd done some cloning in bacteria. We expressed RNA ligase and cloned it. That was a whole nother story that I don't have time to go into. But I thought it ought to go into now, it's time to go into mammalian biology. And just at that point, the HIV program was starting up, the AIDS directed program in the UK. And I started working with Jonathan Kahn, another scientist in the MRC lab, on the HIV, TAT, and REV proteins, which are important regulatory proteins there, and their interactions with RNA substrates, and their binding proteins. Not much was known about that at the time, and we were able to use synthetic chemistry to synthesize the RNAs, sections of the REV response element and the TAR RNA, synthesize the proteins as genes and express them. So we made synthetic genes in the lab, expressed the proteins, showed the binding, and actually that was our first paper in the EMBO journal, in the TAT protein stimulates transcription by binding to the TAR RNA. And we did some mapping of contacts between the REV response element and the RNA as well. So quite a lot of RNA protein interactions, looking to see if we could develop drugs against these specific targets. The whole series of postdocs in the lab here. And John Kahn then at that point founded a small company called RiboTargets in Cambridge to look for small molecules inhibitors of these protein RNA into targets, and also on the ribosome. There were others who joined us, including Venki Ramakrishnan at the time, joined us in this RiboTargets company. However, I wasn't never clear that small molecules would be good for these RNA targets. In fact, it proved to be that it was proved to be much more difficult to define small molecules which would stably interact and block these RNA protein interactions. So the company eventually folded or was taken over by another company. But the idea for me was not small molecules, but oligonucleotides and the antisense area, which to actually block protein RNA interactions. The idea of antisense goes back to Paul Zamechnik back in the late 1970s. And he had the idea to block such interactions by a steric block mechanism. But all the companies which had started in the mid-1980s had gone on to the regular antisense mechanism, which is cleavage by RNase H. However, I thought this was an ideal target to look at steric blocking oligonucleotides, and I teamed up with Jesper Wengel's lab in Denmark to look at locked nucleic acids with 2'-O-methyl nucleotides, and also PNA from Peter Nielsen's lab, also in Denmark. And so we collaborated quite strongly here, and were able to design short oligonucleotides which interacted here, opened up the RNA sequence here, and blocked the binding of the TAT protein, and got actually interference with the transactivation mechanism. And this was a kind of a formative period showing that it was possible here. We also did some work on the ribosome using similar short oligonucleotides. So we started to work on this, but it was clear that if we got some very good results in cell-free systems and showing a blocking of transcription. The problem is now, how do we get these oligonucleotides into the cell? Because this was the key issue which antisense companies were having problems with in the 1980s. And by the mid-1990s, actually, quite a lot of work had stopped, and several companies had given up at this point. And so I thought this was a challenge in the latter part of my career to work on delivery of oligonucleotides into cells. And we started to think about peptides. Having spent some time with Bernard Lebleu in Montpellier, I decided that this was the opportunity to think about delivery using peptide conjugates of oligonucleotides, using peptides as delivery agents. And there was a so-called cell-penetrating peptides, which was starting to be developed at this time. And so Suzanne Perrotte, also from Montpellier, was my first postdoc for this. And I had a series of postdocs looking at the chemistry of joining oligonucleotides and peptides chemically, and we had various techniques here. The simplest turned out to be a simple disulfide joining, and John Turner in my lab developed the chemistry for this. It was actually simpler than anything else. And we were able to make oligonucleotide peptides and show transactivation. And we showed a small amount of activity inside the cell using the sorts of HIV, TAT, and penetrating peptides by this technique. Now, just as a sideline here, 5 minutes, okay. Just as a sideline here, we did some work on microRNA and showed that actually we started to work with quite a lot with PNA. And we showed that microRNA was not we did not require a cell-penetrating peptide to target microRNA. We just needed a few lysines to get inside, and this showed that the microRNAs were not were very accessible and not like the nuclear targets at all. And we did quite a lot of work with microRNA 122 and 155. This was in liver and this was in spleen, but both in cell and also in vivo, showing that the possibilities of using PNA as an oligonucleotide target. So that's just an aside. Going back to the main project, we started to use oligonucleotide peptide conjugates for delivery into cells. And we particularly used, we started with regular oligonucleotides, but actually we got much more success with PNA, with charge neutral oligonucleotides. And particularly with a peptide, this was a transportan cell-penetrating peptide, which gave a little bit of activity. But as soon as we added 6 arginines on the end of it, we had massive activity. So suddenly the arginines allowed us to deliver inside the cell, and particularly of peptide nucleic acids, and we used not only an HIV target, which was our first one, but we started to use Richard Cole's splicing model, because it actually had an upregulation and a much bigger dynamic range in which to use our techniques. And so we were able to block splicing here, and using his luciferase model, we were able to generate luciferase, upregulate it, and using R6 penetrating without any transfection agent, just incubating with cells, and we're able to get into the nucleus and increase the luciferase, showing that we could now have a mechanism of splicing control using these peptide PNA conjugates. Now, I just want to tell you a little bit briefly, but we then teamed up with Matthew Wood in Oxford to look at muscular dystrophies. These are very horrible diseases of young children, the frequency of about 1 in 3,500. This is Duchenne muscular dystrophy. Patients are in wheelchairs by age of 12. There's tremendous degeneration of the muscle fibers. And what dystrophin does, it's a very important protein that anchors actin filaments to the sarcolemma membrane in muscle fibers. And in Duchenne muscular dystrophy, this is there's a genetic deletion here which truncates the protein. So you actually get deletions of point mutations. And it's a very long, stringy gene of 79 exons, and all have to be spliced correctly. And that's what happens. Unfortunately, you don't get dystrophin produced, and you get muscle degeneration. There's Becker muscular dystrophy, which is a benign phenotype. Certain in-frame deletions leading to normal life expectancy. And so Hans van Ommen in the Netherlands had this idea of converting Duchenne muscular dystrophy to Becker muscular dystrophy using an oligonucleotide. For example, if you've got a deletion in exon 50 here, you can't get exon 4950, the correct splicing. So you jump over to the next one, exon 51, and you get an outer-frame messenger RNA, and therefore no dystrophin. Whereas if you can block that splicing event here by an oligonucleotide, then you can jump over that to make an exon 4952 fusion, which is now in frame, and now you get the Becker muscular-like dystrophy, which is very, very mild symptoms. And people have now developed drugs for these, initially 2'-methyl phosphorothioates, but unfortunately those were not successful in the clinic. Tricyclos are being developed by Christian Leumann, which are looking very popular, but the major ones are peptide morpholino oligonucleotides of Jim Somerton, also about 30 years old, as well as PNA is also about the same. And recently, atiplacin, a 30-mer oligonucleotide against exon 51, just the correct sequence, has got FDA approval in October 2016. I like PMO because it's stable to nuclease and proteases, like PNA. It forms strong sequence-specific duplexes. It's non-toxic. It's excreted from the body completely whole, and it's shown in vivo biological activity in many models. And in particular, we can couple to it, just like PNA, some peptides. We did this starting with our R6 penetratin peptide, which we developed before, and now trying to make it protease stable. We introduced some aminohexanoic acid derivatives and some D residues to form our first PIP peptides, PNA PMO internalization peptides. We've published all of this, and these initial PIP peptides showed a higher number of dystrophin-positive fibers than the competitors, the Sarepta companies' peptides. And our collaborators, Matthew Wood and colleagues here, who developed this, we were rather excited that we could now, in vivo, get really good results and delivery with our peptide PMOs. And we first of all had PNA, and then switched to PMO, because actually PMO was much better in muscle rather than PNA. And we've gone on to look at the 5 series. We can now simplify this, so there's aminohexanoic acid and arginines on each side, a short hydrophobic section in the middle. And the exciting thing was that the short hydrophobic region gave us much more activity in heart, whereas arginine regions in general, I will need just one more minute, was very good for all muscles. Heart required this hydrophobic core, and we were able to show that this hydrophobic core is not sequence dependent, but was just needed a hydrophobic core of about 4 to 6 amino acids. These now became important delivery mechanisms, but in vivo, by IV injection into animals. In particular, we have this dystrophic animal called the MDX mouse. And we've gone on to try to develop this into a drug. First of all, with a large company, we licensed this to a large company, Shire Pharmaceuticals, and we developed slightly smaller peptides, PIP9B2, for example, and there was several of these here. However, unfortunately, the company changed management and hasn't developed it. So we were very upset about this. So we decided to beat our own patent and develop our own peptides. And so since then, we've been developing our own peptides, which are not peptides, very similar. I can't tell you about their sequences, but they're arginine-rich sequences, again, rather similar.